Welcome to Genius Gnomes. I'm Aisha Sheikh, and today we are going to be studying the subtopic of microscopy from the chapter Cell Structures from the syllabus 97700 A Levels Biology. So let's get started. So this chapter specifically focuses on the types of microscopes used, which is light and electron microscope, right? And we're going to learn the differences and similarities between these microscopes and what these differences are based off of, right? So before we begin with that, first we're going to be talking about the electromagnetic spectrum and resolution. So what you see over, so we won't be focusing on these other waves as such, but I want you to focus on the visible light spectrum that you can see over here. So as you can see, the visible light spectrum, we can also call this the Vibgyor, right? Violet, indigo, blue, green, yellow, orange, red, whatever. So these have differences in wavelength, right? So as you can see, wavelength decreases as you go from left to right. So red has the largest wavelength, that's around 700 nanometers. Now this you can see, this is nanometers, which is basically a very small unit of measuring something like minute, like it's meters, millimeters, micrometers, then nanometers. Okay, so wavelengths are usually given in nanometers due to how small their size is. So this ranges from 700 to 400 nanometers. Now what are we studying this for what reason? We're basically going to relate this to resolution. Okay, now what is resolution? Resolution is the ability to distinguish between two objects very close together, right? So the greater the resolution of the image, the greater the detail that can be seen. Now for example, if you have a camera with very good lens, then you can also say that the resolution of the pictures it clicks is very high, right? That means that a high resolution means no matter how much I zoom in, or to a particular point when I zoom in, there's going to be great detail that can be still seen. Like it's not going to be blurry and it's not going to be very distorted that I won't be able to tell the difference between 2 points. That's what resolution is, right? We see them we use this term in simple language when we're talking about the quality of a picture, right? So the higher the resolution, the clearer the image, the more detail you can see, and you can easily distinguish between 2 points. If something is something has a very low resolution, then no matter how much you magnify it, no matter how much you zoom in, it's just going to get blurrier and blurrier, and the points will not look separate, they'll look exactly the same. Okay, so that's what resolution is. Now the smaller the wavelength of radiation used, the greater the resolution. So if I were to use the light spectrum over here, and I were to use, let's say, violet light over here, violet light has around 400 nanometers of wavelength, so it's going to have a higher resolution than compared to if I use red light or any other light that has a greater wavelength than violet, correct? Because the smaller the wavelength, the greater the resolution. Another way to talk about resolution is that resolution is half the wavelength of the radiation used, right? So if I'm telling you I'm using violet light over here, which has 400 nanometers of resolution, then 400 nanometers of wavelength, then its resolution is going to be of 200 nanometers. Okay, now what does this mean? If the resolution is of 200 nanometers, this means that objects that are 200 nanometers or greater in size are the only ones that can be seen using that source of light. Okay, so if you see over here in this next picture slide that I have here, we can see that this is a mitochondria over here, which is around 1000 nanometers in diameter. Okay, and over here we have smaller structures that we call ribosomes, which are around 25 nanometers in diameter. Okay, obviously there's a great difference between 1000 and 25. All right, and here we are using a wave of 400 nanometers ka wavelength. Okay, now these waves are passing through these organelles, and you can clearly see that the mitochondria very well interferes with the waves, right? So because it interferes so greatly with the waves, it is very easily visible under this radiation. However, when we're using if you want to view ribosomes, that won't be very much possible. Why? Because ribosomes are only 25 nanometers and they're obviously not interfering with the waves, so they won't be visible under 400 nanometer wavelength of light, right? So what does this tell us? First of all, the resolution is half of the wavelength, which is, if it's 400, then our resolution is 200 nanometers, correct? Second thing that we need to know is objects only 200 nanometers or greater would be visible using that source of radiation. Reason being that smaller structures or anything smaller than 200 nanometers would not interfere with the waves. Okay, then next, now we talk about light microscope. So we know what resolution is, we know what this is based on. So now let's talk about the microscope, right? So light microscopes are the most common microscopes that you can find in school lab, and you've probably used this before as well in your school, right? Now they use light as a source of radiation, obviously, because they're light microscopes. Okay, they use the UV light or any other light source, correct? Now light, we talk about visible light. Now we looked here, the visible light ranges from 700 to 400. You can see over here it ranges from 700 to 400. So the maximum resolution that would be achieved would be 200 nanometers, which was half of 400, correct? Therefore structures smaller than 200 nanometers are not visible under the light microscope. Now another thing that you know when you're using a light microscope is the specimen. Specimen is any kind of any object that you're going to be viewing under the microscope. We call that a specimen, okay? And specimens are usually like a plaster on a slide. Yeah, they're kept on a slide, and they're processed on a slide, and we view that slide in the microscope, correct? So that's what we call specimens, okay? Now most specimens, the cells in the specimen or whatever we are viewing, are colorless, okay? Most of them, most cell contents are colorless. You would not, okay, you can find red in our body because of hemoglobin, it's a pigment, but other than that, if I remove a cheek cell of yours and I view it under the microscope, nothing's going to be visible. Why? Because everything is transparent. Light can pass through everything very easily, and we won't be able to view anything because everything is transparent. You won't find the nucleus being pink and your Golgi apparatus being blue. Absolutely not. You're going to find everything is colorless, and except pigments, obviously, and that light will pass through it immediately because it's transparent, right? So you won't be able to view it as well. You won't be able to see the detail as much as you should, right? So what we do is that we stain these specimens using colored dyes, okay? Simple colored dyes. If you've done this in your school experiments, they'll make you do it, hopefully. You use simple dyes to just stain the specimen, to stain the slide, and then when you view it, there's lesser light that passes through the cell organelles, and it's much more detailed, right? So you can see that some things that absorb the most of the pigment are most visible, and then the other, you can see the borders and everything. It's very clear. It's much more visible under the light microscope. Here are some drawings of what you could see under the light microscope. You can see over here in the animal cell, you can see the nucleus very well. You can see that this nucleolus over here is deeply staining, which means it absorbs a lot of the pigment or the colored dye that you've put, and this is chromat, and you can see another thing I want you to remember is that these membranes that you see, the nuclear envelope and the cell surface membrane, I want you to remember that we can see only a singular line over here. You cannot see anything, no more detail than a single line, correct? It's just like a line. All right, other details that you can see the Golgi very well. You can see mitochondria as very well. Now other than that, everything that's just blank, we're going to label that as a cytoplasm, and then you can see the small granules or small structures here and there. We don't know what those are because it's so small and the resolution is just 200 nanometers that it's so difficult to tell what these small structures are, because a majority of the organelles inside a cell are much smaller than 200 nanometers. So using a light microscope, you won't be able to see much detail, right? So you can just see small structures, but because the resolution is low, you cannot tell what these structures are. Alright, sometimes you can see one big structure, but you don't know what that is because it's just clumped together. Alright, because these things, because resolution is so low that you won't be able to differentiate. You can't tell if those things are the same thing or they're two different things. Okay, that's what you can see as in an animal cell under the light microscope. Over here, there's a plant cell. You can see very clearly there's a large vacuole. It's pretty big, so obviously it's very much visible. You can see the chloroplast over here. You can see the grana inside of the chloroplast. It's just visible, but you can see it. Okay, obviously this is because they have a lot of pigment, right? So it blocks a lot of light, which is why it's very easily visible. Similarly, as the animal cell, you can see the nucleus, the internal structures of the nucleus, the Golgi apparatus, cytoplasm is just the gray area around, and then we can see the membrane, the tonoplast of the small vacuole. Another difference that we can notice is the cell wall. Obviously, animal cells do not have a cell wall. Plant cells obviously do have a cell wall, and there are these connections between cell walls and cell membranes with neighboring cells in plant cells, which is called the plasmodesmata. We'll learn more about this when we go to the subtopic of cell organelles, but right now you just need to know what you can see under a light microscope, how much detail you can see under a light microscope. Now we move on to electron microscopes. So electron microscopes are divided into two types: the SEM, the scanning electron microscope, and the TEM, the transmission electron microscope. Okay, we'll talk about these later, but right now we'll just generalize what an electron microscope is. Okay, so the source of radiation is obviously electron, right? Because light microscopes use light, electron microscopes use electron. Now, how do you use electrons? Basically, you take a metal and you heat it to a very high temperature, and doing this releases electrons from the surface of the metal, and these electrons have a very high energy. They have a lot of energy, and they're extremely excited electrons. Okay, and then you take these electrons and you pass them in a vacuum. The reason you pass these in a vacuum, first of all, because they are so small, they're so tiny. Obviously, do you know how small an electron is? It's then smaller. It's even smaller than the nucleus of an atom. Correct. So it can interfere with air particles. So to avoid that, first of all, we put them in a vacuum. Secondly, so that they travel in one specific path, correct? They don't scatter everywhere. So two reasons we put them in a vacuum is to avoid them from scattering and to avoid them from interfering with other air particles. Now this gives us the most clear and the most focused image we can achieve. Now because electrons are so small, and because they are so tiny and they're so high, they have so much high energy, that their wavelength is around just 0.005 nanometers. That's it. It's just around 0.005 nanometers. And the resolution that is achieved is 0.5 nanometers. Imagine we were using light microscopes, and the light microscope's maximum resolution was 200 nanometers. Over here, it's just 0.5 nanometers. Alright, that's 400 times more than the resolution that was achieved through a light microscope. So this is very helpful when you're viewing details, and almost every other organelle that is present inside a cell can be seen through an electron microscope. Alright. Another point that I had to mention is that because we are using a vacuum, right? I mentioned how we used a vacuum to focus the electrons better on the specimen. We need to realize that water boils in a vacuum at room temperature. Okay, in room temperature it boils. In normal conditions, water would boil at around 100 degrees Celsius. But if you put it in a vacuum, it will boil at room temperature. That is around 23-24 degrees Celsius. Correct. So because of this, we cannot view living organisms under the electron microscope. Basically, something that's already living, we cannot view it, because every living cell, every living organism has some of the other water content within them. They possess some of the other amount of water. We're mainly made out of water. So if you view that under the microscope, we're basically going to boil. So that's not possible. So you'll have to use something non-living or dead. Okay, when you're viewing under the electron microscope. Now we talk about SEM and TEM, scanning electron microscope and transmission electron microscope. Scanning electron microscope is when the electron beam is used to scan the surface of the structure, which is why it's called scanning, right? We're only scanning the structure of the surface. Okay, we're only scanning the structure of the surface. However, in TEM, the transmission electron microscope, electrons are transmitted through the specimen. They pass through the specimen. So whatever we view are electrons that are transmitted through the specimen. However, in SEM, only the reflected beam. So if this is a mitochondria, for example, then if this is the SEM, correct, if I'm using the SEM over here, you can see these waves are coming and then they'll be reflected. So they'll go back. And these waves are the waves that we're going to observe, right? So that way we will be able to see the structure of the mitochondria very well. However, when we're talking about TEM, if this is the same mitochondria that I'm viewing, TEM, the electrons will pass through. The electrons will pass through my Mitochondria are the ones that will be observed, right? And I'll just explain SEM once again. This is the mitochondria, and these are passing. Okay, let's say these pass through, but then there are a few that get deflected and reflected. Okay, some pass back. So these are the waves, that electron waves, that you're going to observe. So this way, using the SEM, you can obviously produce a 3D image, because you're scanning the structure of the organelles, right? You're scanning the structure, because everything is reflected back. It's only the external structure, the surface structure, that you can see, and the image produced is 3D, because only the reflected beam is observed. However, in TEM, because the electrons are being transmitted or passed through the cell, the organelles, the internal structures can also easily be seen, right? The internal structures can be seen, but this produces a 2D image. Okay, now the resolution for SEM is from 3 to 20 nanometers. However, TEM, it's much more. The resolution, the smaller the resolution, remember this, the smaller the resolution, the greater the detail. Okay, so higher resolution is there for TEM because it's 0.5 nanometers. Okay, it's more resolution. You can see the 3D image that SEM produces. I've taken a picture here. These pictures are from the 9700 recommended course book by Cambridge. You can see this the 3D structure that is observed through the SEM, right over here. And this is a cell. This is a simple animal cell that is observed through the TEM. You can see the amount of detail in this cell, right? It's observed, the amount of detail on this image, okay, right? Compare it to what we were looking at when we were looking through a light microscope. This is what we saw under a light microscope. Now look at this. Look at it. I told you to remember the nuclear envelope, right? We can see it as a singular line. Singular line. Yeah, that's it. It's a singular line. Look here. We can see a double membrane. Look at the detail. The nuclear envelope is a double membrane. Before the electron microscope was discovered, everyone thought that it's just a singular line. So it means it's just one, like, it's like a border, like a membrane thing. But no, it's a double membrane. It's a very, very functional double phospholipid bilayer. Correct? So it's very important that you view organelles under the electron microscope to know the greatest amount of detail within them. Alright, we can see the Golgi so well. We can see the endoplasmic reticulums. Earlier, we couldn't even see the ERs. There was no ER in this. There's no ER. It's so small you can't see it. But over here, you can see it so well. You can see every single layer of the ER. Over here, you can see the RER, the rough endoplasmic reticulum, right here. And on the rough endoplasmic reticulum, you can also observe ribosomes, which is so absurd if you think about it, because ribosomes are tiny. But the thing is, electrons are even tinier, so it's so much easier for them to see it. Glycogen granules. These are glycogen granules, right? These are so easily observed under the electron microscope. So obviously you need to remember that you can observe much greater detail under an electron microscope compared to what you can observe under a light microscope, because you're using a different type of radiation which has a much smaller wavelength, hence a much larger resolution. Alright, this is a drawing of what you would see under the electron microscope. Again, you can see the rough endoplasmic reticulum so well. You can see the ribosomes stuck to the RER. You can see the smooth endoplasmic reticulum here. You can see the internal structures of the mitochondria as well. You can see the microvilli, you can see the cell surface membrane, everything. You can see the internal structure of the nucleus also so very well. Okay. Alright. Now we move on to differences between light and electron microscope. Source of radiation, light and electrons. We already discussed this. Wavelength of radiation used. We use I'm talking about minimum wavelength that you can achieve, right? So for light, it's 400. It ranges from 700 to 400, but I'm going to talk about the minimum that you can use. So for light, it's 400, and for electron microscope, it's around 0.005 nanometers. Maximum resolution for light, it's 200, and for electron microscope, it's a 0.5. Lens. Now, light microscope uses glass, and electron microscope uses electromagnetic lenses, right? Because electrons are negatively charged, so you can use magnets to focus these electrons. You don't really need to know this that much. It's not part of your syllabus, the lenses, but it's just a good point to know. Okay. Okay, the more you know. Specimen in light microscope, you can view dead, you can view alive, you can view something with a lot of water content, something with no water content, does not matter. As long as you can see it, as long as it can get stained, you can view it. But for electron microscope, you need to use something that's dead or non-living, right? Because water boils at room temperature in vacuum, so you'll have to use something non-living or dead which does not have any water content. Light microscope, we stain, right? Remember, we stain these specimens. For a light microscope, we stain them using colored dyes. For electron microscope, you cannot use colored dyes because, electrons can't really, there's no specific thing for like viewing color under the light microscope, electron microscope. What you use are heavy metals. So have you seen an X-ray before? That's exactly what it looks like once you view something through an electron microscope. Using heavy metals, you stain something, and basically the organelles that absorb the most heavy metal just appear more darker than organelles that don't absorb so much, right? So it just looks like a black and white picture of an X-ray. However, when you need to produce images like this, you can see this image right here. This is called false coloring. So a computer is used and a few softwares are used to falsely color these images. So if this is something that is the most staining, so I'll just give this color and this, it's up to you. You can decide. You can make a pink, you can make a blue, doesn't matter, right? You just need, because you use the heavy metal, you know that these are different structures because everything has absorbed a different amount of that heavy metal, so you can see that contrast between them. But the original image that you would get would be black and white. And for light microscope, because you've stained the image, the slide before, then you're gonna get a colored image. Alright, now we move on to stage micrometers and eyepiece graticule. So this is an eyepiece graticule over here. This is something that you use to measure the size of what you are viewing, right? The actual size. Okay, people confuse it with the image size. No, image size is once you've printed this thing out. Okay, you've taken a picture of what you viewed under the microscope and you've printed it out, and then you measure the image of that printout. The actual size is what you will measure when it's under the microscope. All right. So we use two things. Well, first we use a eyepiece graticule. Now to calibrate the eyepiece graticule, basically to know what we are measuring is correct, or to know what size of the eyepiece graticule we are using, we need to calibrate this eyepiece graticule. And to do that, we use the stage micrometer. Now usually stage micrometer is already marked, or sometimes this is a random scale, but it's standardized to 0.1 millimeter divisions. Alright, so this over here is a stage micrometer, an eyepiece graticule, and you see this one, the 0.1, 0.2 division scale, that is the stage micrometer. Alright, the eyepiece graticule has much smaller divisions and a lot of numbers in tens, and here it's in 0.1, 0.2, 0.1, 0.2 over here, you can see. Okay, now how do you use these two devices? Very simple. Okay, let's see. Over here I know that this is 0.1 millimeters, correct? Because it's standardized. Stage micrometer, this is a stage micrometer. I'm going to write that as SM. Stage micrometer. It's standardized that it's 0.1 millimeters, correct? And this over here is the eyepiece graticule. Now what I'm going to do is I'm going to see 1.1 millimeter is how many eyepiece graticule divisions. Okay, eyepiece graticule. How many? 10, 20, 30, 40, 50. So that's 50 minus 10, correct? That's around 40 divisions, right? From here to here, we have around 40 divisions, correct? So eyepiece graticule has 40 divisions. The stage micrometer, okay, in 40 divisions, how much? What is the size of the stage micrometer? 0.1 mm. Alright, so if 40 divisions are 0.1 millimeters, one division is how many millimeters? So what do? You do 40 into x, right? Cross multiplication. 40 into x is equal to 1 into 0.1, which is 0.1. And then you find x, which is 0.1 upon 40. This is around 2.5 times 10 to the power of negative 3. Alright, now you have found the size of one division, which is 2.5 times 10 to the power of negative 3 mm. Alright, now I want you to look at this. This over here is a cell. Okay, this over here is a cell. how many divisions is the cell taking up? Okay, from 40 to 60, right? So that's 20 divisions, correct? So one division is 2.5 times 10 to the power of negative 3 mm. So 20 divisions is how many mm? Again, you cross multiply. So you multiply 2.5 times 10 to the power of negative 3 times 20. And what do you get when you multiply that? You get 0.05 mm. So the actual size of this cell is around 0.05 mm. Cool? Okay. Next, we move on to magnification. Now, magnification is obviously how much you've zoomed into your into that cell. What magnification you are, how much you've zoomed in. It's always mentioned in times. You've zoomed in tens, hundreds, thousands, 10,000 times. Alright. Now, when we are calculating anything regarding magnification, I want you to remember this magic triangle over here. Okay, I call this the magic triangle. And why is it magical? Because, okay, you're going to first write down I am this way. And you're going to make a triangle out of it. Alright, very simple. Alright, whatever is on top of each other represents division. So if I'm considering this, that's going to be 1 I upon A. If I'm considering this, that's going to be I upon M. If I'm considering this, it's going to be A into M, where I stands for image size, A stands for actual size, and M stands for magnification. Alright, now let's say I want to find magnification. So what I want you to do is I want you to cover this. Forget about it. Magnification. What is left? I and A. And since they are on top of each other, it's going to be I upon A. So image size upon actual size is equal to magnification. Let's try this again. Let's say I want to find the actual size. I'm going to cover A. What have I left with? Actual size is equal to image size upon magnification. Now let's say I want to find image size, so I'm going to cover image. What do I have left? Image is equal to actual size into magnification. Alright, let's try doing an example. Right, over here we have this question given to us where the actual size of a cell is 20 micrometers. This is micrometers, okay? Micrometers. We talked about nanometers, we talked about micrometers. Basically, there's centimeters, and there is mm, then there is micrometers, then there is nanometers. And when you go from millimeter to micrometer, you multiply by 1000. When you go from micrometer to nanometer, you also multiply by 1000. Okay, easy to remember. Cool. So now let's calculate. What do you have to calculate? We have to calculate magnification. Okay, actual size is given to us 20 micrometers. Image size is 200 millimeters. Alright, 200 millimeters. Now you can notice the difference in SI units. This is in millimeters, this is in micrometers. So we'll need to convert. Whenever you're calculating anything, alright, you need everything in one singular SI unit. Alright, doesn't matter which one. You can keep it in micrometers. You can keep it in micrometers, you can keep it in millimeters. It does not matter. Okay? But you need to keep them in the same SI unit. So they usually, the question when you're asked, they almost always provide you with different SI units, and you have to convert either one of them to calculate. Alright, we have to find magnification. Let's use my trick. We're going to cover magnification. What are we left with? Magnification is equal to image size upon actual size. Alright, what is the image size? Image size is 200 millimeters. I want to convert 200 millimeters to micrometers. So millimeters to micrometers, as I mentioned earlier, is into 1000. So this equals to 200,000 micrometers. Alright, good job. Now we can easily find the magnification, right? Image size. What is the image size? 200,000 micrometers. What is the actual size? Mentioned here, 20 micrometers. So when you divide this, what you get? You get 10,000. Right? You get 10,000, right? Yeah. So the magnification now is times 10,000. Easy, right? Simple. Okay, let us move on. Another question. Alright, let's try this. Now we're telling us to find the actual size. Alright, been given the image size in mm, we have been given the magnification, we have to find the actual size. Alright, let's cover that up. Actual size is covered up. What does actual size equal to? Image size upon magnification. Alright, image size is in 50 millimeters, but the actual size is always mentioned in how much? Micrometers. They ask you, I didn't mention this in the question, but find the actual size in micrometers. Alright, so first we'll have to convert 50 mm to micrometers, correct? So that's around into 1000, right? So that's 50,000 for you. Cool, 50,000 micrometers. Now you can calculate, right? So 50,000 micrometers upon magnification is 2000. The answer to this is 25. So the actual size is equal to 25. What was the unit? Micrometers. Alright. And that is it for this chapter. I hope you understood whatever I tried to explain to you. If you have any feedback for me, please mention that in the comments. And if you want to support me, please subscribe, like, and share this video. And yeah, keep watching. Thank you very much. Bye-bye.